All right, guys. So what we're working on here today is you are going to design a lab activity, okay, that will test the effect of one of the factors we talked about yesterday on the growth of a plant. All right. So what we're looking for here is you come up with the problem, you come up with the variables, you come up with the hypothesis, materials, and then a procedure with which to test it. There will be no analysis, no conclusion, no evaluation because you're not actually doing the lab. All right? You're just designing one. So it shouldn't take too long for us to do that. So a very simplistic one, okay, would be, for example, a problem would be, what is the effect of differing amounts of water on plant growth? Obviously, no one's going to do one that simplistic, okay? But that could be an example, right? The problem would be, what is the effect of water on plant growth? All right. Then your hypothesis would be something like, if plants require water to grow, and several plants are given, several identical plants are given different amounts of water, then those plants receiving the most water will show the most growth. Okay. Everybody with me so far? All right, then you would have your manipulated variable. So the manipulated variable would be the amount of water because each plant in your experiment is getting a different amount. Okay, the responding variable would be the growth of the plant because that's what you're watching for to see what effect water has on it. And then the controlled variable would be, well, there'd be many of them. Anything else that could affect the growth of a plant would need to be kept the same for all of the plants in your experiment. Agreed? All right, and that would be really important. So, and anything else that we talked about yesterday that isn't what you're actively testing would have to be controlled, and you would need to say, things like, what temperature you keep it at, or stuff like that. Okay, soil, all that stuff needs to be considered and mentioned. What are you going to do with it? Then you design. Then you come up with the materials that you're going to need. Okay, plant, dirt, pots, okay, whatever you think you're going to need, right? And then, in terms of, are you going to do this indoors, outdoors, all of that kind of stuff needs to be considered. I'm not really concerned with how much of everything you need, just what you need, all right? So you don't need to, go into detail about, well, how much dirt does a plant this size need? You can just say dirt, okay, and a pot. You can say medium pot or big pot or small pot, whichever, right? I'm not looking for specific volumes and stuff here because we're in a bit of a time crunch to get it finished. Now, the other things you need to do here is remember that in any experiment, you're going to have your experimental subjects. So in the example I gave you, it would be all the plants receiving different amounts of water, right? But I also have to have in an experiment what's called a control group. A control group is sort of the absence of whatever it is you're testing or the natural condition of something. All right, so if I want to find out what the effect of increasing amounts of water is, my control plant needs to be a plant that gets no water. I know that seems really stupid. Everybody knows what happens to a plant when you don't water it. Okay, but you still have to have that. For good science, you have to say, look, plants don't live if they don't get it. Okay, and then you can compare, well, as they got more water, they did better to a point. Then they all started to drown. All right. I mean, you would, in your hypothesis, you said more water means more growth, but at the end, you might find that that's not the case because you drowned 3 plants. Everybody follow? Okay, when water's coming out the top of the pot, it's generally a bad sign. Okay, all right. So those are the things you need to consider. Make sure you've got some sort of comparison control group as part of your procedure. Okay. And the other thing is, the first thing that should appear is an abstract. Okay. Now normally we don't have those on the ones I give you because that's the thing I do with you before the lab when I go through it. Okay. For you, what the abstract needs to be is like a short 2 or 3 sentences about what you know about the growth of plants. All right, so you would say, here's the experiment we're going to be doing. This is what we think is going to be the effect, and here's some practical applications of it. All right, an abstract is basically a way to get people to read your paper. Okay, if it's a true scientific paper, it's a way to kind of get them interested or hooked, especially if you're needing funding for your experiment. Okay, if some company is looking at what you're doing and goes, hey, you know what, if we pay this guy to do this experiment, then we own his results and we can make money off of that. Okay, so it's kind of a sales pitch. That's kind of unfortunately how science works. Okay, you have to sell your experiment to people and then they make money off of it later. Okay, everybody follow? All right, so that's what you need to get started on now. Things to think about in terms of what factors, okay, could be possible. They can be nutritive, okay, which means maybe what you're putting in the dirt or what you're putting in the water. It could be something to do with the light, okay, how much, how many hours, or colors, or whatever. Right? There's any number of things you could try in this experiment. Okay, since you're not actually doing the experiment, you really have kind of free reign. Everybody follow me? Okay, so some ideas there for you to think about. All right, now you have just under an hour, okay, to get that written up. I would like them in the box by the end of class today. Okay, so again, this isn't, I don't expect this to be, university-worthy work, okay, but at the same time, I expect you to do as good a job as possible in the time that you have. All right, so short abstract, 2-3 sentences about what you know about plants, problem that you make up, what's the effect of this on the growth of a plant, your hypothesis, if and then, your variables explained and identified, materials and procedure. That's all. All right? Okay, if you have questions while you're working on it, let me know. MJ, question? Now, the big thing here is this. How many manipulated variables can you have? 1. Okay? You cannot have more than 1 manipulated variable. Otherwise, you won't know which one caused the change in growth of the plants. And this happens to a lot of people when they're designing this experiment. They'll say, my manipulated variable is this, and then in their experiment, they have 2 or 3 things that are changing for all of the plants, and then we won't know which one caused the change. So, for example, if I was testing the amount of water on plant growth, I'd better make sure that all the plants had the same amount of dirt. Because if they don't, then I've also got a manipulated variable in the amount of soil the plants have. And that changes the amount of water that soil can absorb. All right, so I've got one pot that water's always spilling out the top of, and another one that's like a sponge and it's always sucking it up. All right, so you have to make sure you only have one. You're only testing one thing. Okay, does that make sense to everybody? Okay, so be careful when you're designing your procedure that way. Wyatt, question? In the hypothesis, in the then part, yes. All right, but beyond that, no. You can't do any analysis for this because you won't actually have any data. You'll just have the procedure. Okay. Any other questions there, guys? All right, so set to work, okay, and you shouldn't have any trouble getting this done. If you're done, before class is over, and you've got just over an hour, then you can work on some of the other things that you've got coming up due here. Okay? All right, well, let's get to work.